Welcome to a brief look at a recent paper. Once again, on COVID papers, we're going to be talking about the original paper of Sputnik V, the vaccine developed by Gamaleya Institute in Russia. We've seen a massive surge of interest on Sputnik V, the Russian vaccine, this week, mainly as a repercussion of two news. The first one is that India received 150,000 doses of Sputnik V from Russia yesterday, on May 1st of 2021. And the second one, that this week, ANVISA, the Brazilian health agency responsible for authorizing drugs and vaccines, has denied authorization for the Russian vaccine. So combine these two developments along with some smaller news, such as medical tourism events, such as reported by DW News, Germans head to Russia for Sputnik V jab. All of this has led to growing public interest in the vaccine. And by the way, if you're willing to travel abroad to get vaccinated, make sure you subscribe, because my next video is most likely going to be a review of the best options for vaccine tourism. So, starting by acknowledging what has happened this week, we have this news from Reuters. India has received 150,000 Sputnik V vaccine doses from Russia, a spokesman for India's foreign ministry said on Saturday. And we can see them will be sold by Dr. Reddy's Labs, a publicly listed company on the National Stock Exchange of India. By the way, this is absolutely not financial advice, neither is it medical advice. I'm just mentioning information that has been published. And here on The Hindu, we have more details on this vaccine. So on the article updated today, on May 2nd, we can read: India moved a step closer to having a third vaccine in its immunization drive against COVID-19 when the first consignment of Russia's Sputnik V landed in Hyderabad on Saturday. And so by this we can see that India is already using another two vaccines, which now brings the total number to 3. They are Sputnik V, along with the previously implemented Covishield and Covaxin. Covaxin is an Indian vaccine, whereas Covishield is one of the names for the vaccine jointly developed by the University of Oxford and AstraZeneca. It mentions that India has begun the vaccination program for those aged 18 to 44 years old, which is actually pretty younger than many other countries are vaccinating now. And is most likely one of the reasons why we had a massive surge some days ago in searches on how to apply for COVID vaccine in India. Then it mentions, quote, based on the human adenoviral vector platform. And this is going to relate to the question which the Brazilian ANVISA later. The Russian vaccine received emergency use authorization in India last month. The efficacy of Sputnik V has been reported to be 91.6%, and we're going to check this in the Lancet paper later. And here in the end, the RDIF has already formed manufacturing partnerships with at least 5 companies in India for producing about 850 million doses of the vaccine a year. Regardless of whether there are doubts lingering about the Russian vaccine, the way I see it, considering India's current situation, any increased offer of vaccine is to be seen as positive. But as published on India Today, and more relevant to our talks, while there was skepticism over Sputnik V as Russia had not shared its trial data, perceptions changed when the trial data published in the Lancet said the vaccine, quote, appears safe and effective, end quote. And down here, extremely noteworthy, it mentions that, quote, Sputnik V has a regimen with both doses of the vaccine are different from one another. The two doses use different vectors to target the spike proteins of the SARS-CoV-2, end quote. So you can see that a large amount of the interest emanating from India is due to the severe situation faced by the country and the recent arrival of the Russian doses. And now we come to the repercussions from Brazil. ANVISA, the Brazilian agency, denies requests to import the Sputnik V vaccine. Understand the reasons. I'm here providing a free translation of this article from BBC News Brazil. The 5 authors responsible for the technical areas of ANVISA evaluated that the immunizer wasn't able to demonstrate its safety and efficacy. The meeting was concluded around 11 p.m. There was a great expectancy around the decision of the agency because tens of millions of doses had already been bought by states, cities and by the federal government. Brazilian authorities hope that the shipment from Russia would help to alleviate the country's hardship to acquire sufficient doses to increase its immunization rhythm. ANVISA offers many criticisms of the Russian vaccine, for example being denied access to the Gamaleya Institute, as well as pointing out problems that could compromise the vaccine sterility in one of the visited factories in Russia, and questioning the methodology of the study regarding when to perform COVID-19 tests after patients presented symptoms. However, the greatest criticism, and the one that drew the most attention, was the presence of replicating virus in the vaccine. Now, of course, Sputnik V is not composed of coronavirus, otherwise it could actually cause COVID-19, it could cause a disease. Instead, it's made by non-replicating adenovirus, the virus that causes common colds. However, these adenoviruses are altered genetically to produce the spike proteins that the coronavirus uses to enter the cells. That is, without actually being exposed to a coronavirus, you will be exposed to the spike proteins produced by an adenovirus, by a modified adenovirus, and this will cause you to be ready to produce antibodies against a portion of the coronavirus, the spike protein, if you should come into contact with it. The great problem here is that even these adenoviruses should be non-replicating, completely harmless. And as the article mentions, quote, the problem, according to ANVISA, is that the adenoviruses used in the vaccine can replicate. According to Gustavo Mendes, during the virus's culture in the laboratory, they recover their multiplying capability that was detected in all the presented lots. Now, that's not to say that these adenoviruses being infective would cause you to get COVID-19 from the vaccine. Actually, the most likely outcome would be to develop some adenoviral disease. The great problem here is that it allegedly does not comply with the general international recommendations. That shouldn't be the case for adenovirus vaccine. The situation has escalated between the Brazilian Health Agency and the Russian Institute, with increasing accusations and threats, and we will have to wait to see the final outcome. Most likely further studies will be necessary before an objective outcome can be achieved, based on facts rather than just argumentation. However, the great lesson I think we could take from it is asking why a single agency should have all the power to approve or deny the authorization for a vaccine. In a situation of crisis, is it right to deny people the access to try a vaccine even if it is potentially risky? I am, for instance, a physician. I understand the working of the vaccine, as well as its mechanisms and what is a replicating adenovirus. Shouldn't I have the choice to decide if I want to take the vaccine or not, aware of the danger and the risks? Why is a single organization responsible for making this decision for tens of millions of people? But in the end, this brings us to the article published on the Lancet on February 20th, 2021, Safety and Efficacy of an RAG26 and RAG5 vector-based heterologous prime boost COVID-19 vaccine, an interim analysis of a randomized controlled phase 3 trial in Russia, by Logunov, Dolzhikova, Shcheblyakov et al. And you can see from the background in the summary that they had already performed phase 1 and 2 combined clinical trials, and then, quote, here we report preliminary results on the efficacy and safety of Gam-COVID-Vac from the interim analysis of this phase 3 trial. Methods: we did a randomized, double-blind, placebo-controlled phase 3 trial at 25 hospitals and polyclinics in Moscow, Russia, end quote. So, until here, pretty standard, randomized, double-blinded, multi-center, pretty standard, exactly what we would expect. Nothing to critique here. They recruited participants with negative COVID tests. Naturally, they don't want anyone to already have previous immunity, which is the same reason why no other vaccinations in the 30 days prior. And I mentioned that the interval between both doses was a 21-day interval, and that both the first and second doses were adenoviruses carrying the gene for the spike protein of SARS-CoV-2. And then on the findings, they mentioned that overall they recruited 21,977 adults, close to 22,000 people, which were randomly assigned to the vaccine group or the placebo group. Way more people to the vaccine group than to the placebo one. And then the reason this is an interim study and not the full study is because their primary outcome was the prevalence of COVID-19 according to PCR 21 days after the first dose of the vaccine, that is, on the day of the second dose, which means we are lacking the information regarding the protection afforded by the second dose. Still, vaccine efficacy was 91.6%. And then in the introduction, we can read that, quote, adenoviral vector-delivered antigens are known to induce both cellular and humoral immunity after a single immunization, allowing their use as an emergency prophylactics tool in a pandemic. Furthermore, the use of 2 immunizations gives a durable and long-lasting immune response, end quote. So that pretty much justifies why, even if we have such a spectacular result, with a 91.6% efficacy after a single dose, why do we do a second dose? To ensure that the immune response is long-lasting, rather than just immediate. Even though we have a great result with the first dose, it doesn't necessarily mean it's a lasting result. And then, important to our discussion here, they mention that these characteristics make recombinant replication deficient adenovirus RAG-based vaccines suitable candidates for the whole target. So they do mention here that replication deficient adenovirus. And then, quote, although adenoviral vectors might induce immune responses against vector components and attenuate antigen-induced responses, prime boost heterologous vaccination with 2 different vectors allows minimization of these effects. So that's why we mentioned earlier in one of the news reports we saw that this vaccine is actually made by 2 different components. The first dose and the second dose are different. It's a heterologous vaccination with 2 different vectors. One of them is RG26 REG26, and the other one is REG5. That's also, I will have it in the title of the paper, Safety and Efficacy of REG26 and REG5, because it's actually evaluating the combination of these two different vectors used to deliver the same spike information to our cells. That's to minimize the immunogenicity against the vector rather than against the antigen we want, the spike antigen. Now, at the time of the publishing of the data from the phase 1 and 2 of this vaccine, it was very criticized because Russia decided to adopt mass application of the vaccine before the results of phase 3 studies. There is an argument for that, based on the urgency of the situation and the severity of the crisis. But it's interesting to know that many countries waited for the results of phase 3 before approving its use in their respective territories. The participants were stratified by age, and randomization occurred within each strata. This makes complete sense, first, because we want to be able to evaluate how good the vaccine performed specifically in the elderly, for example, which may sometimes be different from the rest of the adult population, as well as we want to avoid the unlikely yet extremely harmful to the research results event in which pretty much everyone assigned to the placebo group, for example, was younger than 40 years old. We want to have it well spread amongst all age strata so we can compare the effects of the vaccine within its strata. They mentioned that both the doses, the first one with the recombinant adenovirus 26 carrying the spike protein, and the second one with the recombinant adenovirus 5 carrying the spike proteins, or the placebo, were administered into the deltoid muscle intramuscularly and with a 21-day interval. Pretty much standard. Then we have a potentially contentious point here, which is they mentioned that subsequent observation visits were planned for day 28, day 42, and day 180. So far we don't have these results in this paper. And that a PCR test was done in combination with the clinical examination on the day of the second dose, day 21. And that during the trial, apart from the screening visit and day of the second dose, no additional PCR tests were done except when COVID-19 symptoms were reported by the patient. Now, an argument could be made here that they should also have tested for IgM and IgG, particularly IgG, because in either group, but particularly in the vaccine group, an asymptomatic patient may have had the infection and no longer be positive for PCR, no longer be shedding virus. This way, if a patient was asymptomatic despite having received the vaccine and was no longer PCR positive by the 21st day, or contracted the disease after the second dose, but presented no symptoms, the study wouldn't show this as an infection because they were not regularly performing PCR tests on everyone or performing IgG tests for immunoglobulins to detect those who had at some point the disease. This is one of the possible targets of criticisms by ANVISA, the Brazilian agency. A counter-argument to this, and one I mostly subscribe to, is that vaccines aren't necessarily designed to prevent infection, but rather, and most importantly, to prevent death and hospitalization. This way, it would undoubtedly be better to know exactly how many cases of the disease the vaccine prevented, but it's way more important to know how many symptomatic cases the vaccine prevented. Preventing asymptomatic cases is useful, but not nearly as important as preventing deaths. So a counter-argument could be made that the biochemical results of the study are not as important as the clinical results of the study. And here we see the study delve deeper into it by saying that blood sampling for assessment of immunogenicity parameters was only done in some study centers selected on the basis of the logistics chain for the delivery of biomaterial to the central laboratory, where primary blood processing was done. And they mentioned that blood samples for antigen-specific IgG analysis are planned to be taken from up to 9,520 trial participants before completion of the trial. Well, this information is welcome. It would be even better if they had the blood from everyone to evaluate IgG. But in any case, once again, perhaps the clinical significance is greater than the significance of the biochemical findings. And now for the big news. On, quote, the day of those 16 COVID-19 cases were confirmed in the vaccine group, 0.1%, and 62 cases were confirmed in the placebo group, 1.3%. Vaccine efficacy was 91.6%, end quote. And so here we have the great results we were hoping for. And you can see that the incidence of COVID-19 was way less in those that took the vaccine than in the placebo group. Second key point, quote, the observed vaccine efficacy was greater than 87% in all age and sex subgroups. Notably, vaccine efficacy was 91.8% in patients older than 60 years, end quote. Once again, 91.8% to 91.6%, it's not statistically significant. However, what's important to note here is that the vaccine works and apparently is safe in the elderly. That's the key point to take here. And finally, what we're just talking about, quote, there were no cases vaccine group and 20 cases placebo group of moderate or severe COVID-19 confirmed at least 21 days after dose 1. Thus, vaccine efficacy against moderate or severe COVID-19 was 100%, end quote. And this is what's really important, not only about this vaccine, the Sputnik, but also about all others. Some of the vaccines seem to be better than others at preventing infection from SARS-CoV-2 overall, although it's hard to verify that because their studies, their trials were performed on different times and different places. However, the most important is that pretty much all vaccines have an efficacy against moderate and severe COVID-19, those cases that cause hospitalization and death, close to 100%, meaning that most likely we can pretty much get rid of COVID mortality and hospitalization with the use of not only this vaccine, but likely all the vaccines currently in production. And then they go on to evaluate the prevalence of IgG in the serum of patients that were or weren't vaccinated, and predictably it was higher in those vaccinated than the placebo group. But I'm not going to go too deep into this. First, because I believe that the clinical results are way more important, reducing hospitalizations and deaths. And second, because the sample was much smaller, 114 for the placebo group and 342 samples for the vaccine group. They also measured the cellular immune response by exposing the patient's peripheral mononuclear cells to SARS-CoV-2 spike antigen to verify the production of interferon gamma. And once again, predictably, they concluded that the production massively ramped up in those that took the vaccine after taking the vaccine, whereas it wasn't high at the beginning of the study and it was never high in the placebo group. Then they move on to safety and adverse events, and show that most adverse events were flu-like illness, injection site reactions, headache, and asthenia. And that 45.3% serious adverse events were reported in the vaccine group, and 23.4% in the placebo group. Technically, the placebo group would be even higher than the vaccine according to this analysis, and that, according to what we've just seen, none of the serious adverse events were considered associated with vaccination. For deaths, non-vaccine related, and pretty much similar results for the elderly. And in the discussion, the study itself mentions what we've talked about. In quote, furthermore, COVID-19 cases were detected through self-report of symptoms by participants, followed by a PCR test. So only symptomatic cases of COVID-19 are included in the efficacy analysis. Then they mentioned that the Sputnik V vaccine has two forms, the liquid and the freeze-dried one, which is easier for transport because it doesn't require temperatures as low as the liquid one. Is the freeze-dried as effective as the other one? Well, I'm not sure this study could be extrapolated to that. However, to some degree, it all depends on the urgency. If you do need a vaccine, whichever formulation is going to be better than nothing. An important yet predictable point here, in this interim analysis we have not been able to assess duration of protection. Median follow-up time was 48 days after first dose. Indeed, we will have to wait for further information on this, how long-lasting the immunity is. And this is a doubt that lingers for all the other vaccines as well, and also for immunity conferred by infection with SARS-CoV-2. And they close the study with some very interesting news, that they are conducting research to investigate a single-dose regimen. So they've said themselves that the reason they do 2 doses, despite the great results of the first one, is to provide long-lasting immunity. Yet they then said in the discussion that we don't yet know how long-lasting immunity for this vaccine will be. So far, we may not even be sure that taking 2 doses of the vaccine will actually substantially increase the length of immunity. So it would be a quite interesting repercussion if we found out that a single dose is enough. It will be a unexpected positive finding. Overall, very important news from one of the most widespread vaccines for COVID-19, published in one of the most traditional medical journals. It's incomplete, sure, it's a partial analysis. We look eagerly for the full picture, including all the results from the second dose and from the other countries. But these are indeed promising results, and we shall see now the further repercussions from it, and whether ANVISA from Brazil will reconsider the decision, or whether further information will be provided to the agency so that it can investigate more deeply the situation. And we shall see if the recent arrival of the vaccine lots in Hyderabad proves useful in improving the current crisis in India. Please bear in mind that this is not medical advice. I strongly advise you to read the study and the news yourself if you intend to make any decision based on it, but beware that both myself and the study are prone to errors. Thank you for watching this video and for choosing to share your time with me. I hope this has proven useful. And subscribe if you are interested in vaccine tourism, because I'm going to try to compile a list of the available opportunities for the next video. Thank you again for your attention, and I hope to see you on the next video.